Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Before we begin, a note from our sponsor. I'm Richard Jacobs, Executive Director of the nonprofit Finding Genius Foundation and host of the Finding Genius Podcast. In late 2016, I was rear-ended at 65 miles an hour by a truck on the highway, which sent me off-road into a ditch. The impact of the collision gave me a concussion and other injuries. At the hospital, a CT scan showed that I had thyroid nodules, which turned out to be cancer. It was then, when I had a biopsy of my neck, that I realized even if I was a millionaire, I wouldn't want a second or a third biopsy due to the pain and the invasiveness of it. And appointments at that time for thyroid experts were 3 to 6 months out. And I was worried about dying now, even if that was irrational. So because of this, I've decided to raise money to conduct a literature review on steroids, on the causes of anxiety and depression, a condition that affects well over 50 million people in the United States and hundreds of millions worldwide. Our goal is to create a codex, a guide that reveals all possible treatments for anxiety and depression for people that live with the condition, or for loved ones that have it, as my wife and my son do. To find out more about our fundraiser, visit findinggeniusfoundation.org and click on Current Initiatives. And now, to our guest.
Hello, this is Richard Jacobs with the Finding Genius Podcast, now part of the Finding Genius Foundation. I have Professor Ailong Ke. He works at Cornell. He's in the Department of Molecular Biology and Genetics, and we're going to talk about his research. So, Ailong, thank you for coming.
Well, thank you for the opportunity here.
Yeah, tell me about your research, please.
Right. So I guess I was trained as a biochemist and a structural biologist. So my main interest is basically RNA biology and trying to understand the role of RNA in biological processes. And so that has been the focus since my independence in 2005. And so there have been two themes going on in the lab. So the first theme was to really understand how RNA could fold into defined tertiary structures and basically confer important functions by adopting that structure. So basically CRISPR-Cas, right? So that has been the focus of the lab for the past 10 years. And so we're trying to understand the mechanisms by which the CRISPR-associated proteins rely on the guide RNA to find its target in a very precise fashion, and then exert, basically nuclease or other activities on the target and destroy the target. And so, as you know, there are a lot of novel tools invented based on this system. And so we're interested in understanding its mechanism and also bring out its applications.
So CRISPR, I know it's a bacterial ability, but when it affects our cells, does it affect just nuclear DNA or does it affect RNA or, how does it work in our cells?
Right. So there are diverse types of CRISPR systems. Turns out that some exclusively targets DNA and others targets RNA exclusively. And now there are systems that turns out to target both DNA and RNA. So just as you can imagine, there are diverse kinds of mechanisms.
Okay. Is there a Cas8 or 7 or is it just 9 and Y?
Yeah, so these are basically named based on the bioinformatics, and so different CRISPR systems have their signature proteins. And so, for example, everyone knows about Cas9, which is a type 2, basically the signature protein in type 2 systems. And there are other interesting names like Cas12. And so in my system, the signature protein is called Cas3. That's the nucleus in our system.
Okay, so again, now that I understand a little bit better, you're using CRISPR-Cas systems to do what? To selectively cleave RNA or DNA, or what's your model and what's your system?
Yeah, so conceptually, this is basically a RNA-guided immunity system, right? So in vertebrates, the immunity system relies on specific protein recognition, right? So the antibodies recognize the antigens. So in bacteria, turns out that there is also a very powerful adaptive immunity system. So basically the CRISPR-Cas, and they rely on nucleic acid-based recognition. So the guide RNA specifies the target based on the Watson-Crick base pairing mechanism, and then it basically guides the nucleus to further degrade the target.
What does that mean, degrade the? I don't understand. What does that mean, degrade the target? What do you mean?
Right. So basically, nucleuses will cleave the target. Right. So most of the nucleuses will basically stop there after they sever the target in halves. So in my system, the nucleus is further fused with a helicase. And so basically, it's a nucleus hooked onto a locomotive. And so together, they will consume ATP and travels on DNA for extended distances. And along the way, the nucleus will keep degrading the target, cleaving the target. And so that will result in a very long deletion on the target DNA.
But what rushes in then to interact with the cleaved pieces or to insert itself in its place?
Right. So basically, it's basically a cleavage, right? So you sever the DNA in half, for example. So in my system, it does that repetitively. So it travels on DNA and it cleaves DNA in halves in multiple rounds. And the after repairing, the end result is a long deletion.
Well, is the cleaving and the repair matched at the same time, or is the repair come a little bit later? And how is the repair accomplished?
That's a good question. So it depends on the system, right? So it depends on whether it's a bacteria host or it's a eukaryotic host. So in eukaryotes, we mostly rely on non-homologous end joining to repair our DNA. So basically, we see two ends, then the enzymes will try to bring them together and immediately stitch them together. So that's the basic repair mechanism. And so from that activity, combined with the CRISPR-Cas3 editing, what we saw are basically deletion editing.
Okay, so again, it cuts the DNA, but does it insert anything, or it just cuts the DNA and the two severed halves are brought together, or what's inserted?
Right. So in most cases, it's a deletion. The default outcome is long-range deletions. And so for insertions, that's an activity that we have to basically couple the CRISPR tools with a template DNA in many cases. And so that's usually not the default outcome. And so for Cas9, you usually need to bring a template DNA and trick the host cells to stitch it in for insertions.
Well, again, what would be the point of mere deletions? Wouldn't you want to insert things instead? Or like, what's the point of the system?
Right. Okay. So you're thinking about applications.
Yes.
Yeah. So again, the application part is the afterthought after we understand the CRISPR systems, right? So the default function of all CRISPR-Cas is to disrupt DNA and basically destroy the pathogenic DNA. And so that's the default outcome, right? So if you were to use it to fix a molecular genetic disease, you have to come up with creative strategies. Usually that's the case, right? So that requires people to think of creative ways. But now, so more recently, there is a new interesting tool. Basically, the transposons will carry CRISPR systems, and together they were able to basically carry out RNA-guided insertions. And so that's a new phenomena, and we're still studying it. So many groups in the CRISPR fields are studying it.
So you focused it all on applications, or you focused on the mechanism of how this works?
Right. So the early batch of people entering into the CRISPR field, most of us were interested in the mechanisms. We're trying to understand. So out of curiosity, we're trying to understand how this RNA-guided system works. And so the application, you could say, is a byproduct of these studies. So more recently, I think a lot of people enter the field purely for the purpose of trying to bring out the power of these CRISPR systems. And so I think my lab is positioned at the interface, right? So we start from understanding the mechanism, and then because we understand our system the best, we're in a unique position to bring out its power for applications. So that's basically how I strategize my research.
Okay. So when you study this in bacteria, how does it work there? What is the model there and the specifics of it? What do they do, the bacteria, when they use this?
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
All right. So the default function for all CRISPR system is basically to use a guide RNA to find the target and then destroy the target or cleave the target, as we mentioned. And different CRISPR systems do it in different ways. And so we focus on the systems that not just cleave the DNA, but also deletes long stretches of DNA. And so it's a fairly sophisticated system. It took us a bit longer to really understand everything in high resolution. But eventually we understood it well enough so that we find the best strategy to bring it into the eukaryotic cells. And essentially, we were able to demonstrate that it works in eukaryotic cells. It worked to a pretty high efficiency. And we show the default outcomes of that editing action in eukaryotic cells, which is basically a long-range deletion.
Well, it doesn't make sense. The bacteria would just delete material out of another bacteria, or they must fill it with something. I mean,
right. So
do
keep
they do
in
it
mind
to themselves? I mean, how does this work in nature?
Yeah. So keep in mind that so this is an RNA-based immunity system, right? So it's immune against, you could say, foreign genetic elements, right? So it touches the theme of basically host pathogen interactions, right? So that's
Does it just cut out endogenizing foreign material, like endogenizing viruses or plasmids?
So the strategy is to identify this mobile genetic elements or foreign genetic elements or parasitic elements, whatever you say it, and basically things you don't want. So use the guide RNA to find those targets and then basically erase it to pieces, right? So not just cut it in half, but erase it in pieces so that there's no chance for that DNA to be repaired, put back together, stitched back together again. And so one could argue that is the most efficient strategy to destroy a foreign genetic DNA. And so that is why, the system, the so-called the type 1 CRISPR is the most prevalent CRISPR system that you can find in nature, in bacteria species. And so it's more efficient at what it's doing, right? So basically to provide the surveillance function to fight off foreign genetic elements.
So for bacteria, it's like a second stage pump. I know they create pumps to pump out toxic material, but if material gets in and then let's say endogenizes into the bacteria's, genetic material, this can cut it out and get it out of there as well, right?
Yeah. So it has to respond fast and it has to respond very efficiently, right? Because otherwise, if the foreign genetic elements gets integrated into the bacterial genome, it's almost impossible to eradicate it, right? So it becomes part of it and it will be too dangerous to cut it out because in most cases, if the bacteria were to attempt to cleave it off, it basically commits suicide. It's not able to repair the genome lesion that it caused on itself.
Okay, so this is a secondary system. Gotcha. So does Cas9 come with it? Do the bacteria simply just cleave out bad genetic elements, or do they put back certain elements, or do they add elements? I mean, what if there's a beneficial plasmid bacteria wants to integrate into its genes? Does it use CRISPR-Cas to put it in there in the first place?
There are different creative ways for the CRISPR systems to be used. Default function is to cleave. Say that's 99.9% of cases, that's what it does. It picks up a new memory, make sure it's immune against that particular phage. Or transposon or plasmid, that kind of thing.
Okay, so you're trying to do this for what kind of conditions? To remove genetic abnormalities, or what's your goal with the research?
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Right. So that is still being developed in the lab, right? So we have a tool that has high impact upon targeting and shreds genetic information into pieces. It's high impact, but it's also harder to tame, I would say, for, say, therapeutic use. And I would say at this moment, it's a little bit dangerous to use as it is for therapeutic usage. And so we, at this stage, we're mostly focusing on the basic science usage, right? So what we're trying to do is to find scenarios where we can apply a deletion editing, generate libraries, for example, and understand what parts of our genome carries out important information. For example, there are many hidden genetic locus that are important for ability of the cell, for ensuring a correct splicing pattern, for gene expression regulation, or even causing diseases. And so it's hard to correlate those kind of locus with the disease phenotype or specific biological processes. And if you were to use Cas9 to screen the specific location of these elements, it will be not very effective and it will be very costly in terms of design the libraries and stuff. But our tool is perfect for that kind of a study. And so we're trying to demonstrate how we could use CRISPR-Cas3 based deletion editing to efficiently zoom into important locuses in the cells that may cause disease or may be very important for biological processes. And so that has been the focus of the lab in the past 2 years.
Okay. So the guiding, I mean, how does the guide happen? How do you get CRISPR to cut out the elements you want? How do you load it up with, how do you know where it's going to cut?
That is the fundamental question that basically inspired a lot of us to dive into the CRISPR system to study it. And different systems does it slightly differently, but the main principle is basically you have the guide RNA, right? So the guide RNA actually can spontaneously invade into DNA because it's thermodynamically favorable. But without the assistance of proteins, this process happens very inefficiently and very, very slow. And so basically that's my understanding, right? So basically what we're trying to understand is how the CRISPR-associated proteins promote the efficient target finding process. Basically, they form RNA protein complexes and they were able to efficiently zoom into the target and then basically form the RNA DNA hybrid and identify the target through that process.
So you say form the RNA DNA hybrid. What does that mean?
Right. So basically the guide RNA has to base pair with the DNA, right? So, and then keep in mind that our DNA is perfectly double-stranded, right? So it's perfectly base paired. And in order for the RNA to specify a target through base pair interaction, you have to unwind the DNA, right? So expose the DNA into single-stranded form and then replace one strand of DNA with the guide RNA to form the DNA-RNA hybrid. And so essentially all CRISPR systems that identify DNA target has to go through this process, unwind the DNA, promote the base pairing between guide RNA and one strand of the DNA, and essentially promote the formation of the so-called R-loop structure. Right, so you loop out one strand of DNA and form RNA DNA hybrid.
So what is it called? Where the RNA and the DNA, the guide RNA first fuses onto the DNA. What kind of feature does it latch onto? Is there a name for that feature?
In order for this process to take place in an efficient fashion, I think all CRISPR systems, DNA targeting CRISPR systems, first search for the so-called PAM, protospatial adjacent motif, or the PAM sequence. Right, so it's going to basically do 3D searching on DNA, look for PAMs, and then when it identifies the PAM, there are plenty of those PAMs in our genome. It's going to try to unwind a small stretch of DNA and see whether it's a good target or not. If it's not a good target, it's going to let go and keep searching for other PAMs. But if it's the right target, you're going to form a small intermediate between the guide RNA and the DNA, and that's going to propagate into basically the entire R-loop region. And the entire process is very much like unzipping a zipper. You're basically unzipping the DNA in a directional fashion, but then during that process, the guide RNA invades and base pair with the DNA.
Okay. Are you able to take CRISPR and modify it to make custom CRISPR that will cut exactly where you want it to cut? Like how would you modify it? Can you?
So people try to do that when they want to enhance their CRISPR tools. For example, the Cas9 is very efficient at finding target, but sometimes it's overzealous at cutting the substrate. So it's the off-target effect is on the high end. So people will come up with strategies to curb its activity and make it more basically a high fidelity enzyme. And so my tool, the CRISPR-Cas3, I would argue it's a pretty high fidelity tool from the beginning. And so there are some inherent features that makes it pretty specific. And so for now, we're not so worried about the off-target effects in our system, but we are interested in engineering the Pam specificity, right? So I mentioned that Pam, all CRISPR tools search for Pam first before it opens the R loop. And so if you want to specify a target, it has to have a Pam nearby, a native Pam. So if there's no Pam, then you're out of luck. You cannot cut that target. And so
can you splice one in one round and then use another CRISPR to go in there and
that would be one thought, right? So that would be one feasible thought. And so, but what people were trying to do is to essentially relax the Pam specificity in their CRISPR tools, make their tool to recognize, additional Pam sequences. And so-called the switching of Pam specificity. And so that could be done through either structure-guided designs or test tube evolution, basically directed evolutions. So both approaches generated very powerful tools for CRISPR-Cas9. And people are trying to replicate the success in other systems.
Okay. Well, what do you think is going to be possible in the next few years, and what's further out versus that?
In general, or in my
with your targeting, with the specific CRISPR targeting with your system, like what right. Hypotheses you're testing right now? What do you think is going to be possible in the next year or so with your research?
Yeah. So certainly we want to make our tool more robust in terms of, for example, relax the Pam specificity so that it can target more, it has a wider target space. And we're trying to tame its deletion activity, so make it more precise. And so the goal is that, for many molecular diseases, genetic diseases, a deletion editing would be perfect as a therapeutic tool. Right. So, for example, if the mutation really messed up the splicing pattern, many ways where deletion will rescue its function, but we have to do it in a very precise and in a very precise way, right? So, in a very safe way. And so that has been the goal in the lab, to find out strategies to make it very precise and no off-target effect, and expand this Pam specificity so that in the end we get a very safe and also robust tool for therapeutic usage in the future.
Okay, well, very good. What's the best way for people to find out more about your research? Where can they go?
Right. So I have a website, and so certainly you're welcome to. So basically, it's a Google website, but if you search my name, you can find it. And I have a Twitter account, and I'm not the most social person. I don't tweet all the time, but I try to be. It seems like the younger generation of scientists are very proficient, more proficient in using those tools than people like me. So I'm trying to adapt to the new waves of communicating with others.
You need to find like a Twitter CRISPR that goes in there and cuts out the information you need and pastes it into the right tweet automatically.
Yeah.
Well, very good. Ailong, thank you for coming on the podcast. I appreciate it.
Sure. Yeah. Thank you again for the opportunity.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.